Hi, everyone. Welcome to Spotlight Session, Lifestyle Manipulation of the Tumor. We have a lineup of fantastic speakers in store for you. My name is Erik Nelson, and I'm an associate professor at the University of Illinois in Champaign-Urbana, and I am one of the moderators.
Hi, I'm Neil Iyengar, and I'm an associate attending at Memorial Sloan Kettering Cancer Center in New York City. And we'll get right into it. We're very excited by our lineup of investigators today. We'll start with Dr. Kristy Brown. Dr. Brown is an associate professor of biochemistry in medicine at Weill Cornell Medicine. Dr. Brown's lab is focused on understanding the molecular link between obesity and breast cancer via the regulation of steroid hormone production by metabolic pathways in breast fat. Dr. Brown, thank you for presenting today.
Thank you so much. I'm going to share my screen. As Neil mentioned, my lab is really interested in trying to understand the molecular link between obesity and breast cancer. And more recently, we've also been interested in seeing how obesity may also impact the development of breast cancer in individuals who have a predisposition to developing cancer. So in particular, women who have a BRCA mutation. So it'll come as no surprise to this audience that obesity is a major health concern. And now with strong epidemiological evidence linking obesity to the development of 13 types of cancer, including hormone receptor positive breast cancer after menopause, there's a real need to identify new strategies for intervention. So in order to start tackling this issue, we have to have an understanding of what drives cancer. And I like showing this sagittal section of the breast because it really emphasizes how much fatty tissue there is in the breast. And also how and where the fatty tissue is located in relation to the breast glands, which are the site of origin of breast cancers. So my lab and others have spent quite a bit of time trying to understand how the factors within the breast actually drive the growth of breast cancers. This includes in response to factors that are produced by the fat cells themselves, the adipocytes. So we know that with dysfunctional adipocytes or with obesity, the adipocytes produce different adipokines, including leptin or higher levels of leptin. We also know that these cells can die with excess energy, and this results in an inflammatory response with macrophages infiltrating the adipose tissue. And these macrophages can produce their own factors, inflammatory mediators that can act not only to stimulate the growth of breast cancers, but also on other cell types within the breast, including the adipose stromal cells of the breast, which we've been particularly interested in, because these are the cells that are responsible for driving estrogen production or producing estrogen within the breast tissue. So we've become interested in BRCA1 and BRCA2 mutation carriers and seeing whether obesity may also play a role in the development of breast cancer in these women. Now, women who have mutations in these genes are typically part of or develop a syndrome called hereditary breast and ovarian cancer syndrome. These two genes will encode two completely different proteins, but these proteins are involved in repairing the DNA within a single pathway, the homologous recombination pathway. So as you can imagine, defects in these genes are actually associated with defects in DNA repair, and as a consequence can lead to the accumulation of chromosomal defects that can lead to cancer formation. And in fact, individuals with mutations in BRCA1 and BRCA2 are more likely to develop both breast and ovarian cancer over their lifetime. There have been a number of studies looking at the impact of obesity and lifestyle on the development of breast cancer in BRCA mutation carriers. A number of them have shown that maintaining a healthy lifestyle or avoiding obesity is associated with decreased penetrance of breast cancer. That said, there have been other studies as well that have not shown this link. So there's still a lot of work to be done to try to understand why there are these differences. And you can imagine that prospective studies are difficult to do because it's difficult to follow these patients for long enough to get a good understanding of the impact of obesity in these patients. And if there is a role of obesity in driving breast cancer in these women, the mechanisms behind it are still incompletely understood. So in order to start addressing this question, we decided that we would test a very controlled system. So we decided to generate a BRCA1 heterozygous knockout mouse in order to be able to study the impacts of diet-induced obesity on the development of breast cancer. Now, these mice were generated on a C57BL6 background, which are more likely to develop obesity. And they are global heterozygous knockouts, meaning that they have a defective allele of BRCA1 in every cell of their body, which is analogous to what you would see in BRCA mutation carriers. So in this case, our mice are ovary intact, again, to better reflect what happens in the female population. And we place the mice on either low-fat diet or high-fat diet. And this is based on 10% or 60% of calories coming from fat. We see that over time, a high-fat diet is actually associated with an increase in weight, which we would expect, but also development of insulin resistance and glucose intolerance. And here we see the data for the glucose tolerance test, where we see a significantly larger area under the curve following glucose administration. In addition to impacts on glucose and insulin sensitivity, we also see an accumulation of mammary fat to a greater extent in the high-fat diet-fed mice compared to the low-fat diet-fed mice. Now, in order to start querying the impact of this diet-induced obesity on the development of breast cancer, we've adapted another model. In this case, we use the MPA-DMBA model to stimulate the development of breast tumors. Here, mice at 4 weeks of age are placed either on low-fat diet or high-fat diet, and at 14 weeks of age are implanted with a methoxyprogesterone acetate pellet, which really primes the mammary gland for the development of mammary tumors. The mice are then given a carcinogen over 3 weeks, and tumor development is examined over time after that. What we found in this study was that BRCA heterozygous mice on a high-fat diet actually had higher mammary tumor penetrance. So by the end of the study, 86% of the mice on a high-fat diet had developed mammary tumors compared to 69% of the mice on low-fat diet. In addition to this, the diet also affected the latency of tumor development. So what we found was that mice that were on high-fat diet actually developed tumors quicker as well. So this was, I guess, the first evidence whereby obesity actually increases mammary tumor penetrance and decreases latency of mammary tumors in this model. But what's the relevance of this to women's breast cancer? And how can we start addressing this question from a mechanistic point of view? So in thinking about mechanism, and because BRCA proteins are involved in DNA repair, we were interested in seeing how diet and obesity may in fact affect the amount of damage within the breast or the mammary gland. So in order to do this, we developed a technique to be able to assess the amount of double-strand breaks or DNA damage in the normal mammary gland of both mice and humans. What we do is we study the phosphorylated form of histone H2AX. Now, this is a histone that is rapidly phosphorylated in the event of a double-strand break by the kinase ATM. And the way that ATM works is that it will actually phosphorylate histones along approximately 100 kilobases on either side of a single double-strand break, making its detection relatively straightforward. So this is an image of an H&E section, and as you can see, the breast ducts are here, and these are the structures that we're really interested in. We're able to stain these with nuclear stain to be able to identify the nuclei of the ducts, but also with gamma H2AX antibodies. And it may be difficult to see, but the result of this staining leads to very punctate red dots throughout the epithelium, which are a direct reflection of a double-strand break. So we use this method in order to be able to assess the impact of various conditions on the integrity of the DNA. So going back to the diet-induced model of obesity on the BRCA1 heterozygous background, and this is not the carcinogen-induced model because we really wanted to see whether obesity was able to cause a change in the mammary gland that would potentially lead to differences down the road with regards to tumor development. So we looked at the number of gamma H2X foci within the normal mammary gland of these mice in relation to their diet. And what we found was that mice that were placed on a high-fat diet had a significantly higher number of gamma H2AX foci, so more DNA damage compared to the low-fat diet. And even though we saw an association with the body weight of the mice, the strongest association was seen when we actually looked at the different fat pads. And when we looked at the mammary fat pad in particular, there was a positive correlation between the amount of DNA damage and the weight of this particular fat pad. So in thinking about our clinical implications, we were able to collaborate actually with Dr. Iyengar at MSKCC to obtain tissue from BRCA mutation carriers. And in collaboration with investigators there, we're able to generate a tissue microarray to be able to analyze a large number of cases at one time. So we were able to study 72 women with BRCA mutations. These spanned a range of BMIs with both BRCA1 and BRCA2 with a mean age of 41.6. And we had both pre and postmenopausal women in this cohort. This is an example of what one of the TMAs looks like. So you can see each individual core is a normal area of the breast. So these women underwent surgery or mastectomy, either for prophylactic or for therapeutic reasons, and we were able to assess the normal areas of the breast here. So the question was, is obesity or BMI associated with more DNA damage in the breast tissue of women who have BRCA1 or BRCA2 mutations? What we found was, once again, a positive correlation between the amount of DNA damage and the body mass index of these women. And this was consistent with the data that we had obtained in our mouse model. So in looking more closely at the data, we also saw an association with age, although this association was not as strong, and with menopausal status, with postmenopausal women having more DNA damage than premenopausal women. So what are the possible drivers of breast epithelial DNA damage? We had a look at a number of systemic factors that may be involved in driving this relationship. And interestingly enough, there was a positive correlation between the amount of damage in the breast and a number of these systemic measures, including insulin, glucose, and HOMA-IR score. And this was suggesting to us that it was not only obesity, but potentially metabolic dysfunction that was driving the amount of damage within the breast epithelium. Interestingly enough, there was no association with C-reactive protein or IL-6, suggesting that inflammation itself is not a major driver of this effect. But what we did see was a positive correlation with serum leptin and an inverse correlation with sex hormone binding globulin, suggesting that higher levels of free steroids were also associated with more DNA damage in the mammary gland. So in order to start asking a question about the mechanism behind this, or which factors specifically might be involved, we decided to turn to our in vitro models. And for this, we started really by looking at what was happening locally within the breast. So in this case, we developed a 3D model system whereby BRCA1 mutant breast epithelial cells, a cell line that's heterozygous has a heterozygous mutation in BRCA1, was cultured in what we call a collagen button. So it's a drop of collagen. And within this drop of collagen, the cells are able to grow into these organoids, if you will, or spheroids, which do end up having a hollow center similar to what we see in the mammary gland. So each of these circles is really a cluster of cells that has proliferated over the course of the experiment. And what we do is we overlay this collagen button with adipose tissue from either lean or obese individuals. And what we found was that if we overlaid the BRCA mutant breast epithelial cells with adipose tissue from obese individuals, we saw much more DNA damage compared to that was overlaid with adipose tissue from lean women, suggesting, number one, that the adipose tissue was able to cause some DNA damage, and that there was a difference between lean and obese tissue. To further ask this question, but in a different way, we obtained conditioned media from a large number of women who had undergone reduction mammoplasty. So from the breast adipose tissue of women who had BMIs from 20 all the way up to 50. And we then incubated this conditioned media with these same epithelial cells. And what we see is, once again, a positive correlation between the amount of DNA damage and the BMI of the women from which we obtained that conditioned media, suggesting once again that the local factors, so the factors that are produced within the breast, are able to cause this effect or this damage to the DNA. So because we had seen a correlation between systemic levels of leptin and the amount of DNA damage, and that leptin is produced locally within the breast, we were interested to see whether leptin may also have an effect on DNA damage directly. So to test this, we cultured our BRCA1 mutant breast epithelial cells in the presence or absence of leptin. And here you see a dose-dependent effect of leptin to increase the amount of DNA damage. When we went back to our conditioned media experiments, where we had seen that obese conditioned media had caused more DNA damage than the lean conditioned media, we were able to reverse the effects of the obese conditioned media when we added a leptin antibody. So this antibody neutralizes the effects of leptin. And these data suggest that leptin, number one, is able to cause DNA damage. And of the factors that are produced by the breast tissue, neutralizing leptin is actually able to abrogate the effects. Now, we were also interested in the steroid hormone question. My lab has been interested in studying aromatase regulation in the breast for quite some time. The reason for this is that aromatase is the rate-limiting step in estrogen biosynthesis by converting androgens into estrogens, and it's a major driver of breast cancer growth, and this is evidenced by the efficacy of currently used aromatase inhibitors. What we had also found previously was that aromatase levels within the breast are positively correlated with body mass index, suggesting that with obesity, we actually have more estrogen being produced that can drive the growth of breast cancers. But could estrogens also be causing an effect on DNA damage? So when we looked at the relationship between the amount of damage in our breast tissue and the amount of aromatase within the breast adipose tissue, there was a positive correlation between the two. And what was interesting was if we took clinical samples, so breast cancer explants from women with a BRCA1 or 2 mutation, we were actually able to reduce the amount of damage by treating these explants with the selective estrogen receptor degrader fulvestrant. So this was suggesting that targeting estrogen receptor action was actually able to decrease the amount of damage within this tissue. So today what I've shown you is really data that we've generated demonstrating a role for the local microenvironment in potentially driving DNA damage and breast cancer development. Now, we knew that the adipose tissue was able to contribute to the growth of breast cancers and breast cancer progression, but very little is known about whether the adipose tissue can actually have an impact on breast cancer initiation. So we're currently performing studies to try to understand how these various factors are causing this damage and whether they're also impairing repair of the DNA, which could also contribute to the findings that we observed. So with that, I'd like to acknowledge the individuals that were actually involved in performing these studies, in particular Priya Bhardwaj. She's been an amazing contributor here. She's led this project and has performed most of the studies that I presented today. She's pretty much in the final year of her PhD, and I know we'll do great things going forward. I'd like to acknowledge others while Cornell, Dr. Iyengar, as I mentioned before, has been instrumental in this study, especially with regards to our clinical samples. Dr. Michael Polak performed the analyses in the blood for us, our funding, as well as women who donated their tissue for these studies. And thank you.
So next we have Dr. Elizabeth Wellberg presenting. Dr. Wellberg is an assistant professor in the Department of Pathology at the University of Oklahoma Health Sciences Center. Dr. Wellberg's lab investigates the mechanisms through which obesity promotes breast cancer progression and the metabolic effects of estrogen deprivation. Currently, she is investigating how growth factor signaling potentiates the response to estrogen in the obese environment through ligand-independent activation of the estrogen receptor. Dr. Wellberg.
Thank you for that introduction, and thank you to the conference organizers and to you, Dr. Iyengar and Dr. Nelson, for the opportunity to share our work. So I'm going to talk also about obesity and breast cancer, but I'm going to focus specifically on the process of weight gain or adipose tissue expansion and how that might promote breast cancer progression. So we all know that obesity is a prevalent disease. This map here shows that most of the states in the US have a prevalence of obesity of at least 30%, if not more. And the proportion of individuals with obesity has steadily increased over the past decades in both adults and in children, and it's projected to continue to increase. So it's important to me that we consider this variable when we're doing clinical studies. So obesity is associated with 13 cancer types now, and among these is postmenopausal breast cancer, which is what I specifically focus on. And the disease of obesity increases both the risk of being diagnosed with breast cancer and also worsens the prognosis for breast cancer. And those two things are illustrated here. This study estimated about a 40% increased risk for postmenopausal breast cancer associated with increase in BMI. And the prognosis is illustrated here in this study that found people with obesity have a poorer disease-free survival compared to those without obesity. And the important thing to keep in mind for what my lab works on is that most breast cancers that are diagnosed in people are ER positive and estrogen dependent. About 70% of them are. So this is the type of cancer that we study. And BMI is what's routinely used to assess population level obesity, but people have begun to ask in recent years if there's a better predictor for breast cancer risk and outcome than BMI. And there are a few things that we can focus on. One of them is metabolic health, which refers to insulin sensitivity and glucose tolerance. Another one is weight gain, which I'm going to talk about today. And this is the process of adipose tissue expansion. And another factor that may be a better predictor of breast cancer risk than simply BMI is adipose tissue distribution. So where adipose tissue is stored around the body, which influences a lot of circulating factors and can influence cancer risk and prognosis. So I'm going to talk about weight gain, which involves adipose tissue expansion and the production of growth factors, which is a normal process. So weight gain specifically associates with postmenopausal breast cancer risk. This report found in postmenopausal women about a 10% greater risk of breast cancer for every 5 kilograms or 11 pounds of adult weight gained. Another study found that looked at weight gain by BMI transition and found that individuals who were normal or overweight as young adults and transitioned into the obese category as older adults had the highest risk for breast cancer compared to all other categories. And I've illustrated this here to show that at the time the study was done, although people may all have the same BMI, the higher risk for breast cancer was seen in people that transitioned from a lower to a higher BMI as they aged. So we modeled this in mice. And as I mentioned a minute ago, the most prevalent subtype of breast cancer is ER-positive breast cancer. And to study that, we felt like we needed to use human tumors, grafted ER-positive tumors. So we use RAG1 null females on the C57 Black 6 background because they're obesity prone. And we mature the females on a low-fat diet or a high-fat diet. And our high-fat diet is about a 40% fat diet. We also use thermoneutrality, which is warming them to reach their thermoneutral zone, which really accelerates weight gain in immune-compromised females. So once they're mature, we overectomize them and supplement them with estradiol in their drinking water, and we implant tumors. And the supplemental estrogen is required to support the growth of human ER-positive tumors, but this process also allows us to withdraw the estrogen without an additional surgical procedure and monitor changes in metabolism and body weight. So once the tumors are established, we simulate endocrine therapy by withdrawing the supplemental estrogen. And I'll explain more about that in a minute. But what we've done is a lot of studies to focus on this period of estrogen withdrawal and have identified that it's about a 3-week period where the mice undergo rapid weight gain that's associated with the loss of estrogen. And this is what we're really interested in. So we've done several studies in this model system, and some tumors that are ER positive progress after we withdraw estrogen, but some don't. And this is really important. So we've used, for example, a PDX that was derived from my former institution, the University of Colorado. It's an ER positive PDX. And when we withdraw estrogen, this PDX continues to grow in the obese animals, but doesn't grow in the lean animals. In contrast, we've also used MCF7 cells, which are an ER-positive line that most of us are familiar with. And these cells don't grow regardless of adiposity when you withdraw estrogen. So they remain estrogen dependent. And here I'm just illustrating the change in body weight that occurs in lean and obese animals during the 3-week estrogen withdrawal period. So the lean animals don't gain weight, but the obese animals do. And of course, we focused on estrogen production by the adipose tissue initially, because there's abundant evidence in humans, including our own evidence and what we just heard from Dr. Brown, that adipose-derived estrogens are an important driver of breast cancer risk and progression in clinical samples and in patients. But we were unable to verify that adipose-derived estrogens were responsible for tumor progression in our model of obesity. And we've used mass spec of adipose tissue, we've used gene expression profiling and ELISA assays of serum. And basically, we cannot detect adipose-derived estradiol in the subcutaneous adipose from lean or obese mice. We can't detect the aromatase gene in those samples. And when we look at the blood of these animals after estrogen withdrawal, we're dealing with values that are right around the limit of detection for the assay that we use. So we were able to rule out estrogen as a driver of obesity-associated breast cancer in ovexed female mice. So we asked, what is driving breast cancer risk and progression? And so revisiting the concept of weight gain, this involves adipose tissue expansion. During a positive energy balance, when nutrients are abundant and the tissue is expanding, in some cases, the tissue produces growth factors to facilitate that expansion in a healthy way. And actually, a paper came out last year, a beautiful paper that showed that hypertrophic adipocytes, these adipocytes get big and they reach a critical size that causes physical stress. And when they reach a particular size, they secrete FGF1. And when FGF1 cannot signal through its receptor in the stromal compartment, the tissue fails to create new adipocytes and grow in a healthy way. So we compared gene expression profiles of breast tumors from patients who are classified as responders or non-responders to aromatase inhibitors. And we cross-referenced that with patients, primary tumors from patients with known BMI. And among the few pathways that were commonly thought to be activated in the non-responders and patients with a high BMI was FGFR1 signaling. And this is consistent with a growth factor-rich environment. So going back to our mouse model, we found that 3-week period of estrogen withdrawal was associated with increased FGF1 expression in the high-fat fed females, the obese females. And again, I'm reminding you here that those were the ones that gained weight. We also found that those high-fat fed females had the largest average adipocytes, particularly after estrogen withdrawal. And FGF1 expression in the subcutaneous mammary fat correlated with mammary fat pad mass, the rate of weight gain in grams per day during that estrogen withdrawal period, and also with the average adipocyte diameter. And we confirmed much of this in clinical samples in collaboration with Dr. Iyengar and his group. So on the tumor side, we found that tumors, this is particularly the PDX that progressed in the obese environment, had elevated levels of activated or phosphorylated FGFR1 in obesity, but didn't have a difference in total FGFR1 levels. In clinical samples, we found that high levels of FGFR1 phosphorylation predicted a poorer disease-free and breast cancer-specific survival in patients after tamoxifen treatment. And in a separate cohort, we found that high levels of FGFR1 phosphorylation were associated with an elevated BMI. So we went back to our mouse model and treated the obese mice who were growing the PDX tumor with an FGFR inhibitor and found that that sensitized the tumors to estrogen deprivation and they regressed. So our hypothesis was pretty simple. Actually, we know that estrogen normally activates its receptor to promote tumor growth, but during estrogen deprivation, adipose tissue expansion is occurring, and the surrounding adipose tissue produces growth factors, which we hypothesize can maintain tumor growth through an estrogen receptor dependent pathway. And this concept of growth factor receptor, nuclear receptor crosstalk is not new. In fact, this is an illustration of the estrogen receptor protein domains and all of the phosphosites and what they can be targeted by, which are a lot of pathways that can be activated by growth factor receptors. So when we're talking about estrogen receptor activation, we're really focused on estrogen receptor phosphorylation, and particularly on the serine 118 protein, which has a very well characterized antibody that we can assay. So the first thing we needed to do was confirm that ER activation or phosphorylation was intact in our model system. And we found that indeed ER phosphorylation was elevated in the context of obesity in our PDX tumors that progressed, and total ER was not different. We also found elevation of ER target genes, PR, TFF1, which is PS2, and GREB1, suggesting that the receptor was in fact activated. And in a clinical cohort, we found that elevated levels of phospho-ER predicted a poorer recurrence-free survival, but total ER levels did not. And this is all strictly ER-positive tumors. So next we went to a cell culture model to experimentally ask if estrogen receptor could be activated by FGF1. And we used all the appropriate conditions of stripped serum and phenol red-free media. And the first thing we looked at was MCF7 cells and the UCD12 cells, which were derived from the PDX tumor. And importantly, I want to remind you that these MCF7 cells were the ones that did not grow in the obese environment when we withdrew estrogen. And here we confirmed that estrogen activates its own receptor, but FGF1 does not. In contrast, the UCD1 or UCD12 cells do grow in the obese environment without estrogen. And we found that both estrogen and FGF1 can increase activation of the estrogen receptor. So one of the questions we had was, is this a PDX versus established cell line phenomenon? And so we analyzed tamoxifen-resistant MCF7 cells, which were derived from this parental MCF7 line. And in this context, under the same conditions, estrogen no longer behaves the way it's supposed to and activates its receptor, but FGF1 does. And we've used a variety of inhibitors to FGFR and a couple of downstream pathways and have narrowed this phenomenon of FGF1 mediated ER activation down to the MAP kinase pathway. And if you've never seen one of these capillary immunoblot systems, this is how we routinely analyze all of our data. And it's really nice because it's quantitative and you can multiplex internal controls and get a really great quantitative measure of what's going on in your cell culture system. So next we ask, what is the consequence of FGF1 mediated ER activation? So what's going on in the cell, and is there something we can use as a biomarker or for treatment purposes? And so we did RNA sequencing analysis of cells at a variety of time points under different treatments, and I'm showing you the tamoxifen-resistant cell data right now. We did pathway analysis of the gene expression signature, and as expected, estrogen treatment makes the cells see that the estrogen pathway is activated, and this can be blocked with ICI, which is fulvestrant, the selective estrogen receptor downregulator. Interestingly, when we treat just with FGF1, these cells also see some evidence that estrogen is there, and part of this can be blocked by blocking the estrogen receptor. And this difference is really what we focused on initially. What is the effect of FGF1 treatment that can be blocked by blocking ER? And these genes fit into these following categories, the top one being glycolysis. Importantly, some of these categories are really consistent with a triple negative breast cancer phenotype, particularly the glycolysis and the HIF1 alpha signaling. And this top pathway, glycolysis, was defined by 6 genes that were really strongly regulated in the pattern that we were interested in. And when we look at those 6 genes in only ER-positive tumors, we see that that signature predicts a poor prognosis for patients in that context. We validated the gene expression effects functionally. Here I'm showing you estrogen treatment, which is in the coral color, compared to the vehicle, increases oxidative metabolism and also glycolytic metabolism. But FGF1 treatment really increases glycolytic metabolism, and this can be somewhat blocked by blocking the estrogen receptor. Those data are graphed here, where we're looking at the contribution of mitochondrial or glycolytic metabolism to ATP production. And specifically, the increase in glycolytic ATP from FGF1 can be reduced by blocking ER. So our working model is really focused on adipose tissue expansion that occurs during weight gain and estrogen deprivation. And estrogen deprivation can occur during menopause, during ovariectomy in rodents, and also during endocrine therapy, which is a really important category of people to pay attention to. And this leads to a positive energy balance. In the setting of a lean, healthy adipose tissue environment, the cells get a little bit bigger, the stromal compartment might expand a little bit, but overall the organism maintains homeostasis. But in the context of obesity, the cells are already stressed, they're already enlarged, and they get even bigger. And then in an attempt to recruit new cells from the stromal compartment, they secrete growth factors such as FGF1 that expands the pre-adipocytes so that they can form new adipocytes. And this process is completely normal, and it's not necessarily something we want to completely block, but it becomes a problem when there's a tumor growing in that environment, such as in the breast, that can respond to these growth factors and proliferate as well. And we think that some of the ways that this happens, maybe one way maybe through estrogen independent activation of the estrogen receptor, and potentially through the acquisition of an aggressive metabolic phenotype in ER-positive cells. So what does this mean for patients? First of all, FGFR inhibitors may be uniquely successful in patients that have activated FGFR signaling. And right now, I think those trials are driven by DNA amplification of the receptor, but there may be other patients who could benefit as well, although standardizing phosphoassays are a bit tricky for diagnostic purposes. Importantly, BMI and metabolic phenotype are not often considered during treatment decisions for breast cancer. There's a lot of gray area here, but we hope that some of our work can help shed light on making that decision-making process a little bit easier. ER downregulators may be more effective in patients with obesity or who are experiencing weight gain, but fulvestrant is really hard on some people. So thankfully, there are a lot of groups working on novel ER downregulators, and we're excited to see what ways those can be applied. And finally, our data suggests that preventing weight gain, maybe without reversing obesity, may sensitize tumors to endocrine therapy. And we actually have a paper coming out pretty soon, hopefully, looking at this phenomenon where we're just blocking that transient period of weight gain where there's a lot of growth factor activity. And I'd like to thank my lab. I have a wonderful lab. I have wonderful collaborators all over the US and in Canada. And I have a lot of tremendous support from funding agencies and from my institution, and I'm happy to take any questions.
All right, fantastic talks so far. Before I introduce the next speaker, I'd just like to remind everyone that after this next talk, there is going to be a lively question, answer, and discussion period, and we request that you all stick around for that and actively participate in it. So with that, I'd like to introduce Dr. Lee Jones, who is a member and attending physiologist in the Department of Medicine and is the chief of the Exercise Oncology Program at Memorial Sloan Kettering Cancer Center. His research program focuses on translational approaches to the investigation of exercise and to inhibit tumor cancer initiation and progression. He has published numerous scientific articles in the area and is a well-respected expert in the field. So with that, Lee.
Thank you, Eric. I'd like to thank the committee for inviting me to present as part of this really excellent lifestyle session. So I'm gonna switch gears a little bit, and we've heard a lot about obesity, and we're going to talk a little bit in this presentation about the other side of the equation, and that's really the role of exercise in regulation of the tumor microenvironment. So as we've heard, we really think about cancer as a tissue-based ecosystem. And in this perspective, we know that, of course, cell intrinsic factors are extremely important for the both initiation and progression of tumors, but this is also heavily regulated by the tumor microenvironment, or so-called extrinsic factors. And as we heard beautifully in the last two presentations, host factors related to obesity can have a significant impact on, particularly in the context of breast cancer, of accelerating tumor initiation and progression. And my belief is this, as Kristy nicely showed, is through regulation of systemic host factors that then have an impact on both tissue or cell intrinsic factors as well as extrinsic factors. So within this context, if we think about obesity and other pathogenic lifestyle behaviors dysregulating the cancer-related ecosystem, that really leads us to what can exercise do to reverse and maybe attenuate some of the effects of pathogenic lifestyle behaviors, with exercise being a physiologic strategy that we believe can influence all the different components of the cancer ecosystem. So as many of you will know, there's now an abundant amount of observational data similar that we have in obesity showing the acceleration of 13 different types of cancer. There's now observational data suggesting that exercise decreases the risk of 13 different types of cancer. And this is illustrated in this figure here. This was a recent paper by Steve Moore in 1.4 million individuals, looking at those individuals reporting high levels of exercise relative to those individuals reporting low levels of exercise. Again, exercise associated with decreased risk of 13 different types of cancer, ranging from a risk reduction of 10% for breast cancer all the way up to about 42% for esophageal cancer. On the post-diagnosis side, which is where we do the majority of our research, there isn't as much epidemiologic research. However, over the past decade or so, an increasing number of studies have now looked at self-reported exercise behavior in a variety of cancer patients, and then risk of cancer-related mortality. And in that context, for patients diagnosed with early-stage breast, prostate, and colorectal cancer, again, those individuals reporting high levels of exercise compared to those reporting low levels of exercise behavior appear to have a risk reduction in cancer mortality of anywhere from 20 to 60%. And this is after controlling for things like age, cancer treatment, body mass index. So this is the adjusted risk reduction with exercise. Of course, we need to keep in mind with this observational data that this is self-reported exercise. It's actually hypothesis generating, it's not hypothesis testing. There's a lot of other confounding factors. Individuals who exercise might eat better, they might smoke less. So I think this data provides a signal that exercise might be affecting cancer pathogenesis, but it certainly doesn't prove that exercise does indeed lower the risk and progression of cancer. To do that, we've switched to mouse models. And over the past few years here, being at MSK, we've now performed a number of mouse experiments. And I'm just going to provide some of the results of these early experiments. So in this series of experiments, these are very simple. This is just using voluntary wheel running as a model of exercise behavior. Essentially, we give access to mice randomized to the exercise group to a wheel. They'll run anywhere from 3 to 5 miles a night. These mice are implanted, orthotopic implanted with various breast cancer cell lines, and then randomized to exercise or not. So here's some of the early data. This is in 4 different mouse models of breast cancer: PYMT, EO771, MET1, and 4T1. And across all these models, compared to the tumor growth rates in the control group, this is depicted here by gray, compared to those who have access to the voluntary wheel, the exercise groups, the significant inhibition or suppression of primary tumor growth across all these different models. And the magnitude of growth reduction is about 30%. And this is just mice receiving normal chow and exercise only. This is not exercise in combination with some anti-cancer therapy. This is just the effects of exercise alone. I think an important question that anyone might ask if you're looking at exercise is, what is the level of exercise I need to do to get this anti-tumor benefit? To answer this question, we then switched to a treadmill model, which allows us to really precisely quantify and prescribe the dose of exercise that a mouse might be getting. So in these particular experiments, these are similar type of experiments. Mice implanted orthotopically with a breast cancer cell line, and then randomized to exercise or not. This time we use a treadmill again so we can deliver a precise exercise dose. We just first look at, this is PYMT, so this is work in a genetically engineered mouse model with mice randomized either to 75, 150, or 300 minutes of exercise per week. The black depicts the control group where tumors appear the fastest. And what you can see with 75 minutes a week, there really isn't much of an impact. 300 minutes is not bad, and there's significant growth delay with 300 minutes, but really 150 minutes appears to be quote unquote the optimal dose of exercise, at least within this PYMT model, to delay the appearance of tumors. In a subsequent experiment using 4T1, which is a triple negative mouse model of breast cancer, this time we tested mice either running 60 minutes, 120 minutes, 180 minutes, or 240 minutes a day, 5 days a week. This is tumor growth over a period of about 30 days. Gray depicts, again, the sedentary control group. And what you can see is these are the exercise doses. And what you can see right away is all exercise doses were associated with significant suppression of tumor growth, but there's no differences between the exercise dose. In other words, 60 minutes a day was just as effective as 240 minutes a day at suppressing tumor growth. There wasn't a dose response effect. So exercise appears to be effective, but there does appear to be this dose threshold beyond which additional doses of exercise don't appear to give you any additional benefit in terms of the anti-tumor effects of exercise, at least confined to these breast cancer models. So what are the mechanisms here? How is exercise related to an anti-tumor growth phenotype within these breast cancer models, or either other solid tumor models? So some of our very early work, and this was in collaboration with Mark Duas when I was at Duke, we were very interested in looking at the tumor vasculature. So this was an early study we did in prostate cancer. These are genetically engineered mouse models of prostate cancer. Very simply, we did some MRIs to look at tumor perfusion or oxygen perfusion into these tumors. And the results were pretty fascinating. On the top panel here, you have, this is the tumor implant. We then did imaging, and these are control tumors. And you can see these control tumors don't light up very much because there isn't much blood flow or oxygen tension in these tumors, because we know a lot of these tumors have poor vasculatures and become hypoxic. But in the exercise conditions, these tumors light up like light bulbs. So we were pretty fascinated by this because, of course, you get an increased blood flow and oxygen into these tumors, but tumor growth rates are slower. We then repeated this. This is Alison Baitos' work that was published in the JNCI a few years ago now. We took this work a little bit further. This is control, this is exercise. So we first looked at the number of blood vessels as measured by CD31. And you can see in the control tumors, really isn't many blood vessels, but in the exercise tumors, there's an order of magnitude greater of blood vessels. We then looked at the functionality of these blood vessels using Desmond, and there's increased pericyte coverage around these blood vessels. So not only are there more blood vessels, but these vessels are more functional. And then finally, we looked at hypoxia within these tumors using both Hooks and EF5. In the control tumors, you can see there's a lot of hypoxia, as you would tend to expect in control tumors. but in the exercise group, really intratumoral hypoxia was significantly decreased. So of course, hypoxia is important for many different phenotypes within cancer. One that we know has come really important over recent years is penetration of immune cells into these tissue microenvironments, these tumor microenvironments, which has been very restricted in a tumor that is hypoxic. So the logical next question was then to see how we can maybe improve penetrance of various immune factors into these tumor microenvironments. So this is not work from our group. This is work from Kristy Brown's lab that was recently published. Some very nice experiments, various mouse models of breast cancer. I think these were subcutaneous injections. And this was, again, treadmill running 5 days a week for about 45 minutes, 7 days a week. Tumor growth across 3 different breast cancer models, all significantly inhibited by exercise after just 7 days. They then did RNA-seq of the tumors. Interestingly, they found increased or upregulation of genes related to inflammation and basically cell mobility, which you would expect the opposite actually with exercise. I'll come back to this a little bit later. But importantly, when they looked at infiltration of CD8 cells, there was an increase with exercise. And when they did an antibody against CD8, this abolished the exercise phenotype. So this is exercise alone with just placebo. You do the antibody against CD8, and the exercise suppression of breast cancer growth is abolished, suggesting that the effect of exercise, at least within breast cancer, at least within some of these models, appears to be CD8 dependent. So the majority of work is clinically, and we felt that the epidemiologic data and the preclinical data really set the stage for us to start to launch some clinical trials to really get to the bottom, I think, the really important question of the biological effects of exercise in individuals and patients with cancer. So I just want to illustrate here in this slide that we've now got a number of studies underway across the entire cancer continuum, from individuals that may be high risk for breast cancer, those with BRCA1, BRCA2, all the way for patients with metastatic breast cancer. This is actually a study run by Dr. Neil Iyengar. But importantly, all of these studies try and take advantage of certain oncology situations where we can get access to either normal tissue or tumor tissue, both before and after exercise training. So we can now start really interrogating how exercise affects the biology of these tumors. Over the next few slides, I'm just going to talk a little bit about one of those studies where we have some pilot data looking at some of these initial findings. Now, to look at the effect of exercise and really test the effect of exercise, it's a little bit challenging, as you can imagine, trying to get individuals to commit to an exercise program that might be anywhere from 6 to 52 weeks in duration, requiring patients to come in 3-5 days a week into a center. You can see that that might be an issue. It's particularly an issue when you live in New York City. Asking individuals to come into the Upper East Side of Manhattan 5 days a week can be problematic. So when we started to design these trials, we knew we had to develop a different type of approach to enable us to do these studies, which didn't involve individuals coming into Manhattan 5 days a week. So we developed something called DigitX. Essentially, we put together, integrated a bunch of different digital platforms that allowed us to do all study procedures remotely. So we would, as soon as a patient come on trial, we would send them a study kit, or we send them a study kit that includes a treadmill, some wireless devices, as well as an iPad. We now can do the consent remotely. Patients don't need to come into Manhattan or any MSK facility to do the consent. We can do a fitness assessment at their home. This allows us now to personalize the exercise prescription. All the exercise sessions are also done remotely with our team of exercise physiologists monitoring these patients in real time during the exercise sessions. Because they can do these sessions at home, now we can blow the doors of the types of doses of exercise we can test. We're no longer restricted to just 3 days a week. And we also, rather than asking patients to come to a center, we actually send a company to the patient's house and allow us to collect samples at a much higher sampling frequency. So as I mentioned, we send patients home with a bunch of wireless devices. This now allows us to phenotype patients in a way that hasn't currently been possible before, whether it's monitoring changes in lifestyle behaviors, whether it's monitoring the physiologic host response in terms of heart rate or maybe even glucose. And of course, in the context of our ongoing studies where we can get tissue both before and after, we can now sequence blood, stool, and also tumor tissue, I think, to get a complete picture of how exercise might be changing the cancer ecosystem or components of that ecosystem. So in the last couple of minutes now, I'll just give you a bit of a flavor of how we are applying this to some of our studies. So we've just completed this study. This was what we consider to be a phase 0 study. Obviously, this is not the first time testing the effect of exercise, but it was the first time that we've used this digital approach to really implement and monitor exercise in any clinical populations. So we took individuals who were preoperative in the window of opportunity between diagnosis and surgical intervention. And we looked to recruit both breast, prostate, and endometrial cancer patients at least 2 weeks between diagnosis and surgery to give us at least some window of opportunity in which to intervene with exercise. And in this first study, it was a very simple exercise intervention, just 5 days a week, 30 minutes each time, which is the current guidelines for cancer patients. So again, just to give you a flavor of what some of this data looks like. So when we give patients some of these wireless devices, now we can start tracking, I think, with really exquisite resolution, the changes throughout a day in terms of things like heart rate, maybe their number of steps. And this is an example of this. So we get a heart rate reading every 10 seconds, 24/7. We have a glucose monitor in some of our studies. This gives us a glucose reading every 15 minutes, 24/7. And so you can see how we can start looking at changes within a particular day. The red bar here represents an exercise session. You can see the corresponding changes, increases in heart rate and steps, as you would tend to expect. But you can do this on a daily basis. Now you can do this over the entire period of an exercise trial intervention. So this is a patient who's on study for about 150 days. Each single line represents an individual day within our studies. The blue represents the sleep patterns, sedentary periods in yellows, active periods, and then the exercise sessions is in red. Again, I'm not really looking to interrogate this data, but simply give you a visualization of how we can use this data to better understand lifestyle changes over the course of an intervention, which can be very powerful when you're looking at exercise or other lifestyle-related interventions. We can also use information now to do longitudinal mapping of host physiological response. This is in one patient here who was on study for 50 days. This is changes in exercise capacity. This is now changes in heart rate, blood pressure, weight, and glucose. So typically studies would just look at changes in heart rate from pre to post intervention, but now you can really get a really beautiful insight into what the longitudinal changes are in some of these parameters. We've also looked at changes in molecular response. Again, this is just for visualization purposes, not for interrogation. So in these patients, we get plasma, we get stool. So now we can sequence the plasma. This is changes in cytokines in just 2 patients over both from week 1, 2, and 3. So now you can look at corresponding changes and how certain factors change over time. We also did metabolomics, so we can now look at changes in circulating metabolites. And then this is also changes in the stool microbiome in patients. So now we can phenotype the host molecular response using a variety of different assays. Of course, within some of these scenarios, particularly within the preoperative setting, we can also get access to the tissue, the tumor tissue. So now we can look at various high throughput sequencing platform to see how we're changing biology. This is just data from some prostate cancer patients in a preoperative setting. This is changes in tumor gene expression. Again, just for illustrative purposes, but it's kind of interesting. Similar to what Rakesh Jain saw in his mouse model, we definitely see this increase or an activated tumor response to exercise, which is maybe contrary to what you might expect. And then we can also do RNA-seq deconvolution approaches to look at changes in immune milieu composition. Again, just for illustrative purposes, this is just across 8 patients, but you can see we start to see some changes in some immune factors, particularly CD4s and maybe some macrophages as well. Again, just for illustrative purposes, but I think now you can start really getting to the crux of the matter of how exercise is changing tumor biology. And then finally, you can now start putting all this data together, which is ultimately what we want to do to really understand exercise in the ecosystem. So I'm not going to go through this in any great detail, but this is the different data types we have, where it's lifestyle, changes in physiology, metabolites, cytokines, the microbiome, or the tumor molecular changes through RNA-seq. And you can see how all this data is talking to one another. So now we think we'll get discovery of not only how all these factors are talking to each other, but also maybe the identification of novel pathways. So just to finish, clearly, I think we know that exercise significantly regulates the host physiology, both on a molecular level and a physiologic level. And I think we're starting to get some insights, both from our preclinical data as well as now our clinical data, that this can have a potent effect on many different tumor biological processes. We've obviously started to look at this in terms of some molecular profiles, but of course, there's a lot of different molecular processes to look at. And I think this is going to be an extremely exciting area of research for years to come. So with that, I close and thank you for your attention.
Okay. Well, that was a really excellent session. Thank you to our speakers for those wonderful talks. We're now going to move on to the Q&A portion of the session, and we do have several questions that have come in. We'll try to organize this a little bit by speaker, but let me use the chair's prerogative, actually Erik's chair's prerogative, because he has a couple of questions for Kristy to kick us off. Erik, I'll pass it over to you.
Sure. Why don't we start with a few lowball kind of technical questions that some of the audience members asked. So Lee, for example, you one audience member wants to know if you could clarify whether you're getting breast biopsies at the residences of the volunteers, or if they still have to come into MSK for the biopsies.
Yeah, thanks, Erik. Hi, everyone. Yeah, so all the study procedures related to the exercise components and consent, everything is done at home in the patient's primary residence, but all the standard of care visits, if it's a biopsy, for example, are still done at MSK. We don't have the ability yet to do biopsies at home. Maybe in the future, that would be something fantastic. I don't know if we're ever going to be able to do that, but maybe one day, but not at the moment. So I guess I should say all study procedures except the biopsy are done remotely.
Excellent. And then for both Drs. Wellberg and Brown, your high-fat diet that you've been using in your studies, is that isocaloric and just higher in the fat, or are there more calories in the high-fat diets?
Okay, I'll get this one started. So our high-fat diet has 60% of calories from fat. It's not isocaloric, but it's not far off. So our low-fat diet has about, I think, 3.8 or 3.9 kilocalories per gram, whereas our high-fat diet, I think, is at 5 or 5.2. There's a difference as far as the carbohydrate, though. So there's actually more carbohydrate in our low-fat diet than there is in our high-fat diet. So that's something that we have to consider in the interpretation of our studies as well.
And our diet as well, our high-fat and low-fat diets are not isocaloric. Our high-fat diet is 40% of calories from fat, and our low-fat diet is right around 10%. And in our high-fat diet, we also add sucrose, which really helps sort of push the metabolic derangements that happen in the mice.
So let me ask a follow-up question to that. Are you able to interrogate how much of what you're seeing is related to, let's say, adipose tissue-derived growth factor secretion versus modulation of insulin signaling, and how much of that may be contributed by the diet through which you're achieving this model?
That's a great question. And so I guess right now we don't have any data to separate those things out, but one of the studies that we're doing that's ongoing is to grow those PDX tumors in the 2 groups that I showed you, but we also have a middle group of animals that's fed a low-fat, high sucrose diet, which promotes insulin resistance and glucose intolerance without weight gain. And we do see that some of the PDXs grow just as well in that environment as they do in the high-fat fed environment, but some of them don't. So I think that it's probably tumor dependent. You know, the studies that we're doing now are going to try to sort out some of the insulin signaling components as well.
Great.
All right, we'll start with some of the deeper questions here. First to Dr. Brown, in your models, so you've observed this DNA damage associated with obesity and aromatase expression, and most of that has been restricted to carriers of BRCA1/2 mutants. Can you expand that to other types of breast cancer? Is it just a general phenomenon?
So we have looked at a smaller population of women who do not carry a BRCA1 or 2 mutation, although there's a caveat there. So we do see higher levels of DNA damage in women who have not been diagnosed with a BRCA mutation. The extent of damage is not as great as what we see in the BRCA1 and BRCA2 population. The caveat is that a lot of those studies that I think that we are hoping to do going forward have been with reduction mammoplasty tissue, and in that case, you know, these are not patients who are actively screened. So, but from the cases that we have looked at in collaboration with Dr. Iyengar, for example, there does seem to be an association there as well, though it's not as strong as what we see with the carriers.
And perhaps dovetailing off of that question, in terms of the DNA damage, do you think that the role of estrogen receptor in the DNA damage is just because of its mitogenic effect, or is there something further beyond the mitogenic effects?
So that's an area we're really interested in as well. And there have been studies showing, you know, effects of, obviously DNA synthesis is associated with a certain level of DNA breaks, as are, I guess, the mutagenic effects of estrogen. So estrogen metabolites, for example, and these can be metabolized in the epithelium to lead to DNA adducts that can lead to damage as well. So there are those possibilities that have previously been reported. In our case, what I can say is that we started addressing that question by looking at the amount of proliferation in our breast glands, so of the clinical samples that we have, and we do not see an association between the proliferative status of women's mammary glands and the amount of DNA damage. That said, it doesn't mean that our in vitro models are not following more of a response to proliferation.
Liz, that question actually came from you. Do you have any other comments or questions with regard to that topic for Kristy?
No, that's, I actually am not very familiar with the estrogen metabolites and their mutagenic roles, which is really fascinating. So yeah, that was what I was wondering.
Great.
Following up on it though, so you also showed that fulvestrant appears protective or can reduce the amount of DNA damage. So I guess how long does a patient have to be on fulvestrant for you to see that effect, or your preclinical models? And I assume that once you have the DNA damage, it's not reversible, although maybe over time those cells will become apoptotic. But any comments you have on that?
So just to clarify, our model is really looking at ex vivo patient samples, and I didn't really go through a lot of detail there, but we basically obtain patient samples from the OR, and then we culture them as explants. So they contain all of the microenvironment of the breast. And these explants were treated for 24 hours, and we saw a significant reduction in the amount of damage. I also have to say that what we're looking at is the double strand break, and that's a snapshot in time, you know, that will be a reflection of, you know, the DNA damage, but also the amount of repair that's going on. And the repair is a normal process that occurs within cells, and you would hope that that happens in an efficient and reliable way, whereby there are no defects that are being introduced. But the problem is with a BRCA mutation, if you actually lose that pathway of repair, you start relying on more or less reliable methods of repair, like non-homologous and joining, and that can lead to more defects. So, we're still trying to figure out how much of this actually, can lead to cancer. And anyone in the audience who knows about fulvestrant will appreciate that it's not necessarily a very good preventative, although there have been other approaches to lower the amount of estrogen that seem to be effective in this population. If you think about oophorectomy or tamoxifen use in a patient who has been diagnosed with breast cancer, in preventing contralateral breast cancer. So there are opportunities there. I think we're exploring metformin. We had previously shown that metformin can suppress estrogen as well as having beneficial metabolic effects. So that might be an opportunity there in the prevention setting.
And Kristy, do you think, oh, sorry, Erik, I'm just going to follow up on Kristy's comment here.
Go ahead.
Sure. So Kristy, do you think that's a unique either mechanism or opportunity for PARP inhibition in the treatment setting that perhaps is not as well recognized?
Well, PARP's interesting, and this is an area that we're also interested in the follow-up of other people's work, actually, who have shown that PARP is also involved in regulating aromatase. So there's an opportunity potentially to inhibit PARP and ACT. The challenge there is that there's still an active copy of BRCA, right? So, BRCA mutation carriers still have an allele with wild-type BRCA. So it's still not clear to us how we're seeing these effects in a BRCA heterozygous population. And we have a couple of avenues that we're pursuing that we think might help explain this, but they're not necessarily getting a second hit right away. So I don't know, the question of olaparib or PARP inhibitors in a prevention setting, it may address that question of, tumors before they're detected as well. I think it's an interesting question.
Yeah, certainly, perhaps limited by toxicity in the prevention setting. Erik, you had a question or comment?
Yes. Jim Johnson in the audience so this is jumping off of your metformin reference. Do you think there's a causal effect of hyperinsulinemia in your in vitro models? And this is for Dr. Brown, but then can easily pivot to Dr. Wellberg.
Yeah, so I think based on our clinical data, so in our clinical samples, we were seeing a better association, I think, with markers of metabolic dysfunction in relation to the amount of DNA damage. And that would suggest to me that, it's not only the dysfunctional adipose, but potentially other factors like hyperinsulinemia that are contributing to this. We have done a couple of studies in that space, and we need to do more to see whether insulin itself can actually be causing this effect. But a combination of all of these factors, I think, is contributing. We're seeing multiple factors, not only one being the culprit here. What's interesting is that sometimes if you target one of them, it's sufficient to reduce the effects on DNA damage. So there may be an opportunity for intervention. As I mentioned before, if we start thinking about treatment or prevention more globally, as opposed to one specific target like the epithelium, the immune system, or diabetes, and find something that acts more broadly, if you think about exercise, or you think about anti-diabetic drugs, or these types of molecules that can act more broadly, we might be in a better situation to prevent and treat the disease.
Great. Lee, I want to shift over to you. We have a couple of questions from the audience for you. First question is, could you talk a little more about what fitness assessments that you're doing remotely, and how are you doing this, and what are the challenges?
Yeah, so once a patient comes on trial, as you know, Neil, we send them a study kit, as I kind of explained a little bit, which includes a treadmill, blood pressure cuff, heart rate monitor, and alike. So, what I presented is taken 3 years to develop. So once we get the equipment to a person's house, we then do a remote call with that individual using the iPad that we send to them. All the apps are preloaded. And then once they have the treadmill and we know everything's working correctly, then for the exercise capacity assessment itself, we'll do age-related heart rate max, and we'll go to about 80% on the treadmill. So these are sub-maximal tests. They're not maximal tests because obviously we're doing it in a remote setting. We obviously make sure that blood pressure, heart rate, other vital signs are normal before we do the exercise test to make sure they're as safe as possible. And then we monitor those, of course, continuously during the exercise test as well. Take them to 80%. At that point, we obviously got time to 80%, and then we'll repeat that, sometimes repeatedly across the intervention, but always as a post-intervention. So we can use that for two reasons. One, to ensure that exercise in the first place is safe. The second reason we use that, of course, is then we can prescribe personalized exercise prescriptions to that individual based on their baseline exercise capacity. And then the third reason is it also acts as a manipulation check, if you will. A lot of our studies now, unlike our older studies, are not directed towards improving fitness per se. But then, of course, if we're going after tissue biology and other changes, and we've seen a fitness response, then we can be pretty confident that there has been a global physiologic adaptation to exercise. And there, I think that puts us in a lot stronger position to interpret what's happening at a systemic or tissue level. So that's the way we do it. And so far, I think it was challenging at first, but within our team, we did as Neil knows, we did a whole bunch of pilot testing with our own team before we rolled this out to patients, and took it one step at a time. But now I think we've got it down, and I think it works very well.
That's great. And a follow-up question, or I guess slightly separate question from Anna Campbell. Are you able to discuss some of your findings, Lee, from the exercise window of opportunity study using that remote model?
Not with Anna, no. I'm kidding. Hi, Anna. It's so we finished the phase 0. It's done. And can I share some of the findings? No, she can email me separately. We're getting pretty close, but it's still very early. We've got a boatload of data to interpret in that study, not just the physiologic changes, but all the molecular changes at both a host and tumor level. All I can say is the data, it looks pretty cool, but we're still working out what it means. And some of the findings are actually a little bit counterintuitive, but I think I can explain them. But yeah, I'm happy. I know Anna's been interested in preoperative studies in breast. Jen Ligabell obviously did a trial in the preoperative setting as well. I think it's a unique setting in which you can look at the unadulterated effects of exercise and other non-regulated therapeutics on tissue biology, tumor biology, without any other form of cancer therapy. The issue, of course, is the short window of time. So this is really what I think prompted us to develop this remote approach, because now we can get a treadmill to somebody within 48 hours, and so we can maximize that window in which to intervene. And so we made that work in prostate, but at least here in Memorial, when we tried to do it in breast, we couldn't make it work because there was, most individuals were diagnosed and at surgery within 2 weeks. Just, we're pretty aggressive, but there's nothing we can do really, I don't think, in a 2-week window. So I know in Europe and the UK, that might be a little bit different. You might have a little bit of a longer period of time, but inevitably, I think you need these remote approaches, particularly to deliver, I believe, the types of doses that we need to have a therapeutic effect.
Great. A question from Ali Gottfried, and I guess this is directed towards Dr. Wellberg. Health at any size is gaining traction in wellness circles. Would this approach be appropriate in postmenopausal obese breast cancer patients with a focus on improving some of the biomarkers you were discussing, rather than focusing on just purely weight loss? And we can use this, with your answer here, to pivot into Dr. Jones had a question he'd like to ask too.
Yeah, and I guess I probably don't know as much about that program as I should, but I do think that, right now, just focusing on obesity and BMI as one thing, one global single disease is causing us to miss a lot of opportunities to help people. And I think that, focusing on metabolic function and tissue, the health of the adipose tissue, for example, or liver function, separately from adiposity or body mass, is going to be important to prevent, cancer recurrence and, initial cancer diagnosis. And I think that goes back to what we were talking about earlier about a lot of the things that drive cancer are complex whole body mechanisms, and sometimes just blocking one or two can really have a big impact. So I think that I like seeing the merging of the endocrinology and the oncology fields the way that they're coming together right now.
So that leads into the larger question. Do any of the panel members really believe that we can offset the adverse pathogenic effects of obesity for breast or other cancers, say, for example, using just anti-VEGF or FGF therapy? Or what do you think is really needed to achieve the goal of outweighing all these negative attributes of obesity? And I should put a little star, with the exception of immune therapy in obese melanoma patients, as we heard about earlier this morning.
Isn't that fascinating? Yeah, I think, we've kind of talked about this as a group, and so I'll let the others talk a little bit, but I think that we just need to get past the obesity as one thing. Obesity as a single disease, kind of like we did for breast cancer. Breast cancer is called one thing, but there's, as we all know, there are lots of different kinds of breast cancer. And I think that once we know more about obesity, we can intervene a little bit better.
Kristy, do you have any insights on this topic?
Yeah, well, they're very much my opinion, but I think the challenge is, and for most of the epidemiologists as well on this call, is, how do you define how much obesity and for how long is important here? And there have been so many studies, and the metabolic effects probably more so than the actual BMI. But and as a basic scientist, you like to think, well, the research that you do may be able to contribute to our understanding of this. And I think, the challenge for us is that we're looking often in our BRCA population at premenopausal women. We're seeing more DNA damage in these women. There have been studies suggesting that obesity at any point in someone's life increases the risk of cancer. So is this, really generating these tumor cells relatively early on, or, at an earlier stage in someone's life, whereby interventions may be effective in the sense that they may start reducing the factors that stimulate the growth of these cells. So once they've established, can we actually keep them in check? Which I think is what we do for most breast cancers anyway with breast cancer therapy. So by reducing those pro-proliferative factors that we know are elevated in obesity, to stimulating potentially factors that can suppress the growth of cancers, and Dr. Jones alluded to exercise being able to do that. So potentially exercise, changing healthy adipokines, for example, maybe these factors can keep things in check. So I think that, having a healthy lifestyle at any point in your life would be beneficial to prevent or to decrease the risk or the progression.
Great, and maybe related to this, this question is for Dr. Wellberg. You showed nicely in your models the activation or the generation of a growth factor rich microenvironment with weight gain. Have you been able to identify a threshold or even a dose response relationship, if you will, that perhaps could translate to humans as we think about not just metabolic phenotypes, but the natural history of the development of obesity?
That's a good question. I have not been able to do that, but I think about that a lot. So, the lean animals gain weight as well during estrogen deprivation, but they don't produce the FGF. And so part of the basic research in that, at least with regards to FGF, has to do with the physical tension that a very large adipocyte is experiencing. So I think we've tried to look at adipocytes of different sizes to see if we can, identify a threshold, like you said. But yeah, I think that the other part of that is that some of our studies that we've done on subcutaneous adipose tissue from lean and obese mice. We've done single cell sequencing, and I think that the cells are just really different in the context of obesity between lean and obese. So it's hard to know whether they would, I guess one of the thoughts we had is, would the obese cells produce FGF even under the same size conditions as a lean animal, because the cells themselves are different? And that's a bigger challenge than, it makes it harder to reverse those things and prevent the growth factor environment if the cells are fundamentally different. So yeah, I guess that I don't really know the answer to your question, but it is something that we're thinking about and trying to use our models to understand better.
Following up on that, I guess two follow-up questions. The first one is from myself. How chronic is it, right? So if you get these mice obese, they're spewing out FGF. In your preclinical models anyways, if you stop with the high-fat diet and they maybe start losing weight, can you reverse that phenotype, or they just, once it's there, it's always up?
That's a good question. We have not done weight loss studies in these mice yet, but I will say that the FGF seems to really jump during the rapid weight gain during estrogen withdrawal. So it isn't really much higher under the estrogen supplemented conditions. In between lean and obese, it's like this: the estrogen withdrawal, the rapid weight gain, adipose tissue expansion really bumps it. So I don't know that it sticks around for very long, but probably every time that weight gain happens, it just probably floods the system with more growth factors. But the weight loss question is really interesting. It's something we want to see how persistent those effects are, for sure.
Sure. And could you, from Catherine Cook in the audience, wants you to comment quickly on other contributors to FGF, like fibroblasts?
Oh, yeah. We don't see much of it expressed in the stromal cells, at least in our single cell sequencing data. And that includes a lot of different fibroblasts, pre-adipocytes. So I know that, fibroblasts produce it, but in our models, it seems to really be from the mature adipocytes specifically. But that's a good question.
Great. Well, I think that is all the time that we have for this session. So I'd like to thank all of the speakers for their excellent presentations and all of the audience members for their excellent questions. We look forward to the subsequent sessions as part of this conference. And thank you again. Have a great afternoon, everyone.
Thanks, everyone.